Share screen. Okay. So where we left off, we were talking about irradiation, irradiating these raspberries. And then we talked about filtration, how we mechanically separate the microbes from the liquid because these pores in the membrane of the filter are too small to allow them to pass through, but they're just small, but they're just big enough, of course, to allow the water to go. Okay. So what we're going to finish up with today is looking at things called germicides. To review, we've been talking about disinfectants. Disinfectants are things that don't completely sterilize. They're microbicidal because they kill microbes, but they don't kill the endospores. And disinfectants are things you use on inanimate surfaces, not on living things. Unless you're testing them on lab animals, of course, for like cosmetics and stuff. But antiseptics, those are going to be the things that are like disinfectants. They don't necessarily kill off everything, but they're applied to animate objects, to us. Sterilants kill off endospores and everything else. So you have a completely sterile piece of equipment or media or whatever you are sterilizing. And then we talked about degermers, preservatives. Like degerming isn't microbicidal, right? It's more microbial stasis. And so what that means is that it's just preventing the bacteria from, or whatever microbe, from expanding its population, from growing up more. Okay, it limits it. Now, when we're going to use germicides, there are certain things we want to know about them to decide which is the best germicide to use for the particular application. And some of the things that make germicides particularly useful is that they can kill microbes pretty quickly in low concentrations. Okay, so when you're using these germicides on your hands, for instance, you don't have to have a high concentration of it, which could be potentially damaging to your tissue. They need to be often soluble in water or alcohol because microbes are going to be sufficiently hydrated. And so you want to deliver it in something that is also of a liquid milieu. It makes it easier to apply it. Broad spectrum. So what does that mean? Broad spectrum. Well, we're going to talk about that in the next chapter. But broad spectrum means that it kills a wide range of microbes. So you have narrow spectrum and broad spectrum. Broad spectrum is going to kill off a lot of different types of microbes. And in doing so, it's going to have low toxicity to the host, right? And that's the goal, is to kill off the microbes and not harm the host. They have penetrating factors, so if they can be applied topically, but then can be absorbed internally to get microbes that are invading beneath the skin. They're non-corrosive and non-staining, which is important, right? As we said, when we're looking at things like surgical equipment, you don't want it to rust, right? So you don't want to corrode these materials that you're using. You don't want it to stain, right? I mean, that would just be a disadvantage if it stains whatever materials you're using them on or your skin. And ideally, they're going to be affordable and readily available. Although, that kind of was not the case when COVID first hit, as you guys, I'm sure know. Not only was toilet paper in high demand, but things like Lysol wipes and hand sanitizer for a little while were kind of hard to come by. Okay. Germicides have a range of efficacy in what they kill. So we have high-level germicides. These are going to be things that can kill endospores, all right? So they're sterilizers. And these are good options for treating devices that aren't able to tolerate high pressure and steam and temperature in an autoclave, for instance. Okay. So like plastics, but need to be sterile because they're going to be used in a human, for instance. So like if you're applying surgical gauze inside an incision to sop up some blood, you don't want to be putting gauze in there that is not sterile, right? Because then you're introducing bacteria or other microbes into a place where it's supposed to be sterile. So these, but you can't really, I guess you could, I guess you could autoclave gauze if you put it in a bag. But anyway, point being, there are some things that need to be sterile but cannot withstand autoclaving. And so that's why these germicides come in handy. Intermediate level, they're going to kill fungal spores maybe, but not endospores. And remember, fungal spores are different from endospores. Endospores are bacterial. It'll kill things like tuberculosis, certain viruses, and these are going to be used to disinfect equipment that may come into contact with mucous membranes, but aren't invasive. Okay, so maybe dental equipment, things like that, that are going to be used in your mouth, but aren't going to be making incisions. And then you have low-level germicides that will take care of some vegetative bacteria. Remember the non-endospore version, and some viruses. And this is going to be things that come in contact with your skin, like, you know, your hands, but not necessarily inside your mouth. Any questions on that? Okay. What we're going to do now is we're going to go into these different germicides and talk about them in a little bit more depth. When you select a certain germicide, you need to look at different factors, such as what is the material that you want to treat. Okay, and that's going to dictate which germicide is going to be effective or not effective. How much contamination is in that material, right? If it's heavily contaminated, some germicides may not effectively eradicate that contamination. How long do we have to expose the material to that? Okay, so for instance, if you're applying betadine to skin before you draw some blood, for instance, you really don't have to do that very long. I don't know exactly how long. It's like 30 seconds or a minute, right? And it's effective. You don't want, if you're going to be taking a blood sample from somebody, you don't want to use a germicide that requires 5 or 10 minutes to do its function, right? Because that's just inefficient. Concentration of the chemical agent, right? So things that have to be more highly concentrated are probably not going to be better candidates for using on human tissue than things that have less concentration. And we determine that concentration by dilution factor, parts per million, or a dilution percentage like 70% ethanol or 90% isopropyl alcohol, that kind of thing. These are the germicides, and be familiar with these different germicides. You don't need to know every single thing that we talk about with these germicides, but be familiar with these different varieties. First, we have the halogens. And if you guys remember from chemistry, the halogens are right next to the noble gases. And in case you don't remember halogens, I've worn my nerd shirt for you. So right here, oh, there we go. This is the noble gases. They don't react with anything. They're too good for other elements. And then right next to them are the halogens. So fluorine, chlorine, bromine, iodine, and so forth. Okay. Phenols, that is a particular type of organic molecule. And if you're taking organic chemistry, you're probably familiar with phenols. Chlorhexidines, alcohols, hydrogen peroxide, aldehydes, gases, detergents and soaps, certain heavy metals, dyes, and acids and alkalis. Okay, those are the major categories. And some of these should be pretty familiar to you. Growing up and just being alive, you get a cut. Chances are maybe you put some kind of alcohol on there or hydrogen peroxide. Although technically, you really shouldn't use hydrogen peroxide on cuts because it is a radical and it will attack your tissue too, and it will kind of slow down wound healing. But it's okay to use on equipment. Like if you need to get a splinter out of your finger and you're using a needle, then you can put the hydrogen peroxide on the needle. Okay. Any questions so far on this? No? Okay. So halogens. Think chlorine, right? Hypochlorites, that's going to be bleach. How halogens work is that they denature proteins by disrupting the disulfide bonds. Remember, sulfur is a necessary bioelement, and it allows proteins to have their particular structure by forming these bonds between two sulfur atoms. Okay? And a conformation, a specific shape of a protein confers a specific function. And if you denature, meaning if you change the shape of that protein, it no longer does its function. So that's how these halogens work. They're intermediate level. Oftentimes, they're not going to be stable in sunlight, okay, which is why bleach and stuff is usually in not a clear container. And it's going to be used for treating water in your pool or hot tub or sewage. So unfortunately, we're not going to be touring the stupid facility here. But I always do, and I have previously, because they use microbes as a part of the process to decontaminate and break down certain organic matter in the water that they get. And it's a really cool tour, and I feel bad that that I can't do it with you guys. But the last treatment before they pipe all of our water in Tahoe, all of our wastewater actually gets piped up over Luther Pass, and then put into the valley down there as irrigation water. But before they pump it up over the pass, they treat it with essentially bleach. Okay. Iodine or betadine is more commonly used for topical treatments on us. So it could be in some kind of therapeutic ointment, or it can be, like I said, used to put on the skin to decontaminate it before you're going to stick it with a needle or make an incision. Here are some examples. So using chlorine in this chlorine bath to treat cantaloupes that often are harboring different microbes on the outside of them. And so you don't eat the rind, but what do? You cut open a cantaloupe. And remember, these microbes are everywhere, and it's very easy. As soon as you put that blade on the cantaloupe and you start cutting, now you have bacterial cells on the blade, and then you go into the fruit, and now you've just seeded that fruit with all the bacteria on the outside of the rind. So they'll give them these chlorine baths to kill off the particularly bad stuff that causes foodborne illness. Here's a patient being treated with like a betadine solution to decontaminate the outer skin before surgery. Phenol is kind of nasty stuff. So it is sometimes used topically. But it does have a high degree of toxicity. So more common, it's used on devices and equipment. Um, what this does is it's going to disrupt the cell walls and membranes and then precipitate out the protein. So the protein all clumps together. And in the next chapter, we're going to go into the different ways that um, microbicides work on germs, right? It can denature protein that we talked about with the halogens. It can break down the cell walls and membranes that are the protective, selectively permeable outside coat, right? And then there are other things that can get involved. Certain microbicides can mess up the metabolic processes that are necessary for life, and we'll get into that next chapter. But again, these phenols are also intermediate level, bactericidal, fungicidal, virucidal, but not sporicidal. Okay, so that's why it's intermediate, not high level. One example of this is triclosan, which is it's the component that is an antibacterial soap that really none of you should be using, because it's a great way to facilitate antibiotic resistance. Yeah, and like I said, they're toxic, so we typically don't use them as antiseptics. Chlorhexidine, so also denatures the proteins that are necessary to keep the germs alive. It is a more broad spectrum germicide, so it can kill a lot of different critters. But it's low to intermediate level. Okay. And this can be used on the skin before operation. It can be used on burn victims, because if people have had extensive burns, that is now destroying your protective barrier and lending yourself to all kinds of infections now. And so when you want to apply something to those burns, it can't be something that's going to, be very irritating to their already traumatized tissue. So this is something that works well without being super, super uncomfortable. It's in mouthwash, so it's a mild germicide in terms of its toxicity. It doesn't have an effect on the host, but it does work to some degree on germs. Alcohol, right? You go into a doctor's office, it always has that smell, right? That isopropyl alcohol smell. Okay. That's because that's a very common germicide used in hospital and doctor office settings. It's intermediate, and typically it's higher than 50% concentrated, usually like 70% for alcohol, ethanol. Ethanol and isopropyl alcohols are both alcohols. They just differ in the number of carbons. And again, if you're taking OChem, you'll know the difference. 2 carbons for ethanol and 3 for isopropyl alcohol. And so these are going to kill living cells, not living cells, vegetative cells, and some fungi. And the reason you don't just use 100% alcohol is that there needs to be some amount of water so that it can do its job, okay? Because what this is doing is it's going to denature and conglomerate the proteins of that bacteria or fungus together so that they can't do their function, okay? But to do that, it's got to enter into an environment that is mostly water, right? 70% water. And so that's why using alcohol that has a certain degree of water is more effective than using just 100%, because then that talks about the penetration effect, right? And getting to where it needs to go. Hydrogen peroxide. Again, it's a radical, so it's going to damage protein and DNA. It's going to attack the structure, the molecular structure of these very important biomolecules. Okay, it's going to take off hydrogens, and when you do that, when you take off something, even if it's a little hydrogen, you change that molecule. It's no longer the molecule that it was. And when it's no longer the molecule that it was, it can't do the thing it's supposed to do. Okay. So hydrogen peroxide doesn't denature proteins like halogens. It doesn't conglomerate them and precipitate them like alcohols. What it does is it just attacks them. It takes a little piece out, right? And all that little piece is very important. At very low concentrations, you can use it as an antiseptic, right? But again, I wouldn't use it on wounds because it's not great for the healing process. There are certain contact solutions that have hydrogen peroxide. And a little story of mine, I wear glasses, obviously, and I do wear contacts sometimes. And I just use the regular contact solution that's not hydrogen peroxide. For a while, my wife was using the hydrogen peroxide solution, which is a pretty high concentration. But then before you put them in this little chamber, you squeeze in the hydrogen peroxide, and it's much more effective at disinfecting your contacts than just the regular solution. But there's a critical step where before you put them in your eyes, you take another solution to neutralize the hydrogen peroxide. And I didn't know she was doing this, and so I just grabbed the contact solution thinking it was the same stuff that I was using and put it in my little contact case. And then the next day we're at my in-laws' house, and I put the contacts in my eyes and shrieked very loudly, and it was very painful, and I was embarrassed as a result of that. But so hydrogen peroxide is good, but not good in your eyes. So just leave it at that. Aldehydes. These aldehydes are things like glutaraldehyde and formaldehyde. And these are things that are going to be high level, okay? They alkylate protein and DNA. So alkylate mean they stick on a group, an alkyl group specifically. And so when they do that, they, as a result, change the structure, right? And now that structure is altered. It can't do its function. It can be used as a sterilant, okay? So this is a high-level germicide. It can sterilize things. It can even take apart the endospores for heat-sensitive instruments. Formaldehyde is also anywhere from intermediate to high level, and this is particularly used to fix things. So if you are taking a specimen and you want to fix it in a solution so that you can then slice it and put it on slides or dissect it, but you don't want it to break down, you use formalin, which is just a percentage, a 37% solution of formaldehyde. And what that does is that cross-links all of the macromolecules so they don't break down, because they're now all linked together strongly. So gases and aerosols, also alkylating, also high level, also not used on people, also used on devices that can't withstand the heat of an autoclave. So plastics that would melt, right? A lot of prepackaged foods or surgical equipment are going to be sterilized using this type of germicide. So when I'm talking about these germicides, and what I want you to know, do I really care that you know that ethylene oxide or propylene oxide are these gases and aerosols? Not so much. I want you to know that gases and aerosols, for instance, are high level, right? And what are their applications? They're used to sterilize plastics that can't withstand autoclaving, right? As opposed to things like alcohol and some halogens like iodine that can be used on directly topically on people. Okay, that's the kind of resolution I'm looking for. Detergents and soaps, right? These are things that are going to be very low level. They sometimes they can alter the membranes of the bacteria and the fungi, and those are going to be your detergents. And that's what separates a detergent from a soap, right? For the most part, because they can get into the membrane. Soaps are alkaline, right? And if you guys have watched Fight Club, you know all about how to make soap with fat and lye. And so it's basic, and it removes dirt and grease, right? Because grease is just oil. And it removes the grease and the dirt that contain the microbes, right? So when you wash your hands, you're not killing the bacteria or the fungus. You are simply just removing them from your you're removing them from the surface of your hands. Okay, so these are going to be very weak germicides, and only really are effective at killing very sensitive bacteria. Okay. Sorry. Okay. So, concept check. Which of the following kills by producing oxygen radicals? What did I say was a radical?
B.
Hydrogen peroxide.
Bingo. Yes, hydrogen peroxide. If I said something like, which germicide is more effective when diluted in water as opposed to not being diluted, fully concentrated? Which one would you say?
A, alcohol.
Exactly. Okay, that's kind of what I'm looking for. Heavy metals. Okay. Silver, mercury. So thiomersal is a mercury component that was in some vaccines, that is used as the explanation for why vaccines are toxic. However, they're not in any vaccines. I don't know if they're in any vaccines now, but they're certainly not in any vaccines that are administered to pediatric patients. And it's always about the dose, right? And the vaccines that did or do contain thiomersal have a dose of mercury that is so low that you get exposed to higher concentrations of mercury just being alive. So anyway, won't beat that topic down. But point is, these heavy metals in low concentrations can inactivate proteins. Okay, so this is a low level germicide. And what you see here, this is a silver amalgam. This is gold foil. And then what you guys will hopefully see in the lab this Friday is you'll see this halo around it. That's called the zone of inhibition. That means that these bacteria have all died off where you put that germicide, and nothing can approach it. So anytime these bacterial cells divide and they move outward, they get immediately killed in proximity to this heavy metal. Okay. Any questions on that? Okay. I think we're almost done here. Yeah, we got 2 more. Dyes. Aniline dyes can be effective against gram-positive bacteria and certain fungi. Sometimes it can be used as an antiseptic on wounds, but it's a low level, narrow spectrum, right? Okay, so against some gram-positive, and we know there are gram-positive and gram-negative, so it really is not a broad-acting germicide, and it's not a very effective germicide because it's low level. Acids and alkalis, also low level. Certain acids can prevent the spore from germinating. Okay, so it just effectively cuts it off at the metabolically inactive stage. Acetic acid inhibits bacterial growth. And interestingly, acetic acid is another byproduct made with the SCOBY, which gives it a slightly, so if you are making kombucha and you put it in a jar, then you can't put a lid on it because you need gas exchange. It needs to get oxygen. So it's not going to be carbonated at that point because the CO2 is going to come out. And if you let it go too long, it will eventually break down all the sugar, and then the taste will be very tart or sour because there's no more sugar left. It's all acetic acid. But you can leave that SCOBY in a jar for a long time. And if you have good sterile technique to begin with, you really won't get much contamination because of this acetic acid byproduct that's made, interestingly. So acetic acid is a pretty decent and effective way at limiting contamination in your kombucha. And some people drink it and they think it has beneficial properties that way. I don't know. I don't think there's much science on that, but propionic acid slows mold growth. You might remember propionic acid from the first Journal Club paper that we read. Lactic acid prevents anaerobic bacterial growth. Benzoic acid and sorbic acid inhibit yeast. Also, benzoic acid is applied topically to treat acne and things like that. And then that's it. That's all of chapter 10. So, any questions on that? Before we move in, we got about 10 minutes to dive into chapter 11. And then we'll take a 10-minute break and we'll do Journal Club. Okay. Okay, so unless there are any questions, I'll just jump into chapter 11. Okay. Let's see here. I'm looking at the chat. Not the only one making Fight Club connections. That's good. But I would, if it, yeah. So Sean, I wouldn't expect any less. So, okay, sorry. Let's do this. All right. So we've been talking about microbicides and microbiostasis techniques, germicides. Now let's talk about antimicrobial drugs, antibiotics, right? They're different. right? It's a medicine. So it's going to be different than alcohol or bleach, right? Despite what our president says, don't consume disinfectants, okay? Don't shine light inside your body. Those are things that aren't meant to be inside of us. That's why they're different from antibiotics and other antimicrobial medicines, okay? Difference. I don't have to tell you any of this, but you know, based on what's out there in the world, you never know. Okay. So what are we going to be looking at in this chapter? We're going to look at drug discovery a little bit, right? You guys know that penicillin, we talked about that, how that came from a yeast, kind of serendipitously. I think we talked about that the first week. Most antibiotics come from a natural source, bacteria and fungi that are secreting these things, not because of us, but because of other microbes, right? They're all living in the same area and they want their territory. They want their little niche. And in order to get that, they have to kill off the other microbial invaders, right? Other antimicrobial drugs are synthesized. We've made them chemically. Some are originally come from natural sources, but then we genetically engineer bacteria to produce those antimicrobial drugs. There's a little bit of both, right? We initially get it, like a lot of medicines come from plants, right? We initially isolate the compound from a plant, but then we make it synthetically in a lab. Okay. We'll talk about do no harm, right? That's the Hippocratic Oath. And you want to have drugs that are selectively toxic, that kill the bug, but not the host, right? Unfortunately, with cancer treatments, like with chemotherapy and radiation, that's not as dialed in. But with our antibiotics, we've got a pretty good system where, you know, unless you're consuming alcohol or doing certain things while taking the medication, it shouldn't be too toxic. Okay. We're going to talk about how antimicrobial drugs work. All right. And there are 5 major ways that they do so. And you will need to be familiar with these different ways. Okay. We're going to be talking about these 5 ways throughout this chapter. Okay. Some of them inhibit the cell wall. So that means that they prevent the cell wall from maintaining itself. Some disrupt the cell membrane. So that's not that they prevent it from maintaining itself, but they just break it down. Others get in the way of DNA and RNA synthesis. A 4th way is by getting in the way of protein synthesis. And that's done several different ways. And then finally getting in the way of metabolic pathways so that they can't make the metabolic byproducts or the metabolites that they need to sustain themselves. Okay. We'll get a little bit into the side effects of different drugs. You don't want to have drugs that disrupt the normal microbiota, but unfortunately that happens, right? You take an antibiotic regimen and that's not going to just kill the bad stuff. You also don't want to introduce something that's going to be an allergen to the patient, right? Some people are allergic to penicillin. Interestingly, a lot more people think they're allergic, but actually aren't. Ryan, are you allergic to penicillin? Is that one of the many on your panel? Or are you free? Are you okay with penicillin?
Dude, I'm indestructible. I actually don't know. I'm not a big penicillin eater, so I couldn't tell you. I mean, we could test it, I suppose, at some
Yeah.
point.
Okay. And that's exactly the thing, right? Most people who say they have a penicillin allergy, they do so because they were told that by their parent. And maybe they did have some adverse effect, but it wasn't the penicillin. And then they never get tested, but then they just harbor this idea that they have an allergy or a sensitivity to it, when in fact, they actually don't. Some people are allergic to iodine, right? My wife's allergic to iodine, so she can't eat any shellfish, unfortunately. And we don't use iodine tablets to decontaminate our water when we're out backpacking. So you got to think about which things are going to be effective at killing the microbe, but not harming, inconveniencing the host. We'll get into antibiotic resistance. What causes it? Well, spoiler alert, natural selection. These things evolve very quickly. And because they evolve quickly, they can change, not actively, right? Evolution doesn't work that way. It's not like, oh, I need a longer neck, so I'm gonna start getting a longer neck so I can reach those leaves. No. It's mutations that happen randomly that then have some benefit. And so because they benefit the host, that host survives and reproduces. And by reproducing, it passes on that beneficial mutation to some. And to others, maybe it doesn't. And the ones that don't have it, they die off. But their progeny that do have that advantageous mutation carry on, right? And we'll talk a little bit about that as well. Yep, what happened? There we go. I think we'll leave it at this, actually, guys, because there's only a couple minutes left. So what that means is that for the quiz on Friday, you'll only need to know chapter 10 stuff. Okay. This chapter we'll save for another time. Okay. Let's take a 12-minute break and come back at 11. I'm excited to talk about this paper. I'm curious what your thoughts are. I liked it, but we'll see how you guys feel. So let's take a break. We'll come back, stretch your legs, get a drink, go to the bathroom, grab a snack, all that stuff, and let's get ready to talk some yeast. Okay. All right.